We're talking about trying to retrain the immune system so we don't have to just reduce the immune responses to anything, but just trying to reduce the immune responses to the organ itself so that we can re educate the immune system and mitigate the side effects that the immunosuppression drugs come with.
A donated organ can save your life, but from the moment it's placed inside your body, your immune system sees it as an invader and tries to destroy it. For decades, the solution has been powerful anti-rejection drugs. But what if there's a new way? If we could teach the immune system to make peace with a transplanted organ instead of fighting it? That idea once seemed impossible. But now scientists are looking to the body's own playbook, unlocking the liver's unique ability to promote immune tolerance and developing therapies designed to teach the immune system acceptance rather than suppression. That's what we're talking about in this episode of Tomorrow's Cure from Mayo Clinic, a podcast that brings the future of medicine to the present. I'm Lindsay Sievert. It's great to have you with us. Joining me are Dr. Timothy Tanner, a transplant surgeon at Mayo Clinic, and Dr. Angus Thompson, a transplant immunologist at the University of Pittsburgh Medical Center. Dr. Tanner and Dr. Thompson, it's so wonderful to have you both here for this conversation today. And before we get started, I heard you talking earlier, and I understand that you're longtime colleagues. Tell me a little bit more about your capacity of which you've worked with one another.
Yeah, thank you very much, Lindsay. It's an honor to be here. We go back quite a while. Angus was my PhD advisor and mentor and has been my mentor ever since. I owe him a lot to what I became today.
For me, it's been extremely gratifying to see Dr. Tanner flourish in his career, becoming a leading internationally recognized surgeon scientist who contributes continually to organ transplantation. It's always been a great pleasure to have been his early mentor and to remain friends for many years.
Well, that is just wonderful. And before we get into this conversation, I just love to set the stage since you both specialize in transplant medicine and started all those years ago. So for listeners, when they think about transplant medicine, many might just think about just the surgery itself. Many don't know that transplantation is really the beginning of a long relationship between a patient, a new organ, and the immune system. And for more than 50 years, transplant medicine has relied on this trade-off. You get a new organ in exchange for a lifetime of medication. But researchers are chasing what many call the holy grail of transplantation, a future where some patients may no longer need anti-rejection drugs at all. So that's what we're diving into in this episode. And Dr. Tanner, can you just give us a short summary of the reason why we're talking today?
We're talking about trying to retrain the immune system so we don't have to just reduce the immune responses to anything, but just trying to reduce the immune responses to the organ itself.
Dr. Thompson, how would you give us your elevator pitch of that same advance?
As Dr. Tanner has said, we are currently highly dependent on anti-rejection drugs that are non-specific in how they affect the immune system. It's rather like a blanket suppression of responses to all foreign entities. Where the field is going is towards selective suppression of the anti-donor response. And these approaches show significant promise in being able to instill unresponsiveness specifically to the donor, which would be of great benefit to the patients in reducing their dependence on non-specific immune suppression and eventually becoming independent of these anti-rejection medications.
While advances in medicine explore these different kinds of cell therapies, they all share the same goal: teaching the immune system how to accept a transplanted organ. That way, patients may someday rely less on lifelong anti-rejection medication.
Just to give you a scope of the issue, in the United States, about 50,000 people get an organ transplant every year. So if you add this up, it adds up to 500,000 people in 10 years. In the world, we're approaching 180,000 transplants a year. So this is the scope of the problem. This is a problem that the modern medicine created. Transplant was not available 50 years ago. Now it's readily available. It's one of the most successful treatments that we can offer.
So, Dr. Tanner, at what point do immunosuppression drugs come into the picture? And can you help us understand why they are such a central part of transplant care?
Immunosuppression drugs come into the field right away at the time of the transplant because the immune system works in a way that it's designed or evolved to react to anything that's not a self. Any different organ coming from a donor in a recipient's body will be seen as a foreign. It's got to have a lot of proteins that are seen as a foreign. So the immune system starts recognizing it within minutes and starts attacking it. So the immunosuppression medications are there to suppress that response, to modify it so that the organ does not have a rejection. If you don't control this, is what we call the rejection.
And that is a reality for many patients that do experience rejection or they can.
Correct. So unless the immune system is adequately suppressed, people will have rejection. Rejection may sound like a scary word, but once detected and treated adequately, it usually does not lead to long term consequences. But it needs to be treated with, most of the time, enhancing immunosuppression, increasing the dosage of the immunosuppressant medications.
And what does that look like for, let's just say, a liver transplant recipient? Is it a daily pill? What kind of medication is it?
So, let's say a liver transplant recipient, they start with a regimen of 3 different immunosuppressant drugs. They are all pills. They take this every day. And usually, because of the liver's unique tolerogenic ability, we're able to reduce that to just one drug, one at night, one in the morning after the first few months. But we don't have that luxury in other organ transplants. Most other organ transplants, including kidneys, lungs, or hearts, people may need to continue taking 2 or 3 different medications throughout their life.
And these medications can have some tough side effects in some cases.
Correct. These drugs, as effective as they are, unfortunately come with some side effects. Correct. People may have increased incidence of infections, increased incidence of some certain types of cancers, and they also increase the incidences of diseases like diabetes and hypertension.
So let's say that someone receives a new organ. At what point do those anti-rejection drugs come in? Is it immediately?
It starts immediately. Whenever we use an organ for somebody else, the organs, we all have different genetic makeup. So we all have proteins on our organs that are seen as a foreign by the recipient's immune system. So you have to train the immune system not to see that protein as a foreign antigen. So you have to start that at the very beginning, right at the time of the transplantation.
Dr. Thompson, we've all heard the stories of people waiting for an organ for so long. What do people misunderstand about anti-rejection medicines?
It's important for people to recognize just how much enormous progress has been made, certainly in my lifetime, from considering transplantation as something that really was not possible to something that has become relatively straightforward standard of care procedure. I think people do appreciate that immunosuppressive drugs are effective. They do recognize that there are certain untoward effects that can accumulate over time. It's remarkable that we are dependent on the same armamentarium of drugs that we've been using for the past 2 to 3 decades or even longer, and that advances towards the holy grail of being able to minimize, discontinue anti rejection drugs over time has proven a really tough challenge. Although many advances continue to be made that give us a great deal of hope that we will, in the foreseeable future, be able to get larger numbers of patients on very low doses of anti rejection therapy and even off all immunosuppression altogether.
What is the holy grail? I heard you say something, the idea that someday a patient will receive a transplanted organ without being on lifelong immunosuppression medicine. Is that what that is?
Well, as the Holy Grail was a term that was coined many years ago by immunologists and surgeons, and it's the vision that a state of transplantation tolerance can be achieved in patients. And by tolerance, the patient is able at some stage post transplant, usually many years, to become a potential candidate for withdrawal of all immunosuppression. This is possible in liver transplantation, where, depending on length of time post transplant, it may be possible to cautiously withdraw immunosuppression from the patient and the patient does not reject. But that has not, as I mentioned earlier, been an easy thing to prospectively undertake.
Dr. Tanner, that holy grail, the best chance of studying that holy grail or getting closer is, I heard Dr. Thompson say, is within the liver. So what work is underway at Mayo Clinic where you're studying this closer?
Yeah, so before that, let me tell you why the liver we think is special, because liver is, through the evolution, just trained itself to not react to these proteins, these antigens, because it's constantly exposed to dietary antigens. So whenever we eat or intake something, it goes through the bowel, it gets absorbed, and all that nutrients, they get into the bloodstream, and that blood goes through the liver. So liver is trained to see those antigens and not react to it. So it's just one of those evolutionary things that allowed us to come this far. Because of that, through many years of research, we know that the liver transplant recipients, just as Dr. Thompson said, are probably the first cohort of transplant recipients that we can try to achieve tolerance in. We have done several trials trying to get or induce tolerance in liver patients, and Dr. Thompson has done a very innovative trial at his place using those. The trials we have done at Mayo Clinic are those that involve infusion of cells into the recipient called regulatory T cells. So these are lymphocytes or white blood cells that are in charge of controlling their counterparts. So they are the gatekeepers, so they control the other lymphocytes from attacking the liver.
As researchers at Mayo Clinic and the University of Pittsburgh test new approaches, they work towards the same goal: retraining the immune system so transplant recipients could one day face fewer side effects and a better quality of life. Has this been a problem that you've been trying to solve as clinicians since you worked in Dr. Thompson's lab?
I completely agree with Dr. Thompson's sentiments about the immunosuppression drugs and how it made us this successful. We recently had a 50-year celebration of our abdominal transplant surgery division, and looking back, it gave us a chance to reflect on the progress we made. In 1975, and kidney transplant recipients had about 30% of expected 1 year survival. And we are now talking about over 99% thanks to these anti-rejection medications. However, they do come with certain risks. Recently, the American Transplant Society did a survey in over 10,000 transplant recipients, and 92%, while they were very grateful and happy with their progress, stated that they had at least one side effect of these anti-rejection medications.
So, Dr. Thompson, tell me what's underway at your lab with this research.
For many years, this goes back to when Dr. Tanner was doing research in Pittsburgh, and earlier, we've been interested in a particular type of immune cell, actually different from the regulatory T cells that Dr. Tanner has described. The cells that we are focused on are not lymphocytes, but innate immune cells of the myeloid, not the lymphoid system. And these cells have the ability to very effectively regulate immune responses. So, in the trials that we have been conducting in live donor liver transplantation, we generate these regulatory cells from the prospective liver donor and we infuse the cells before transplant or after transplant into the liver recipient. We follow the patients for a year. We perform what is called a protocol biopsy 1 year post transplant. And if the patient is clinically stable and has no evidence of any ongoing rejection, we can cautiously begin to withdraw immunosuppression. And in some patients, we have been able to withdraw immunosuppression completely. And then in following these patients, they have remained off all immunosuppression for 3 or more years post-transplant. So we are encouraged by, first of all, the feasibility of doing the study, the safety of the cells when infused into the patient, and what we would call exploration of efficacy. We have some encouraging signs that this intervention may be successful. However, we have to now undertake what's called a randomized trial to formally test efficacy of the cell therapy approach to improving outcomes.
When researchers take specialized immune cells from the organ donor and give them to the transplant recipient before or after surgery, again, the hope is that these cells help recognize the new organ as something it doesn't need to attack. The science is still evolving, but the promise is significant. Researchers are now working to confirm those results in larger clinical trials. What is the breakthrough that allows some of the successful patients to withdraw from their immunosuppression drugs altogether?
These donor-derived cells express the crucial donor antigens that are recognized by the recipient before transplant. And we believe that the cells bearing those donor antigens that also have an immune regulatory function teach the recipient's immune cells to be less responsive to the donor organ when it's transplanted into the body.
Dr. Tanner, what is the breakthrough here for you and your work?
This all shows me that we're getting one step closer every time we talk about this. There are several approaches. Dr. Thompson's approach has been very successful. At Mayo, we are also trying to retrain the immune system of the recipients. Another way of doing this is trying to achieve what's called a chimerism. So if you can retrain the immune system by introducing the donor's immune cells into the recipient and they start to live together without attacking each other, then you can put that donor's organ into the recipient without having to worry about rejection because you had already retrained it. So that is done through infusion of stem cells that are obtained from the recipient. All these kind of approaches, including the regulatory T-cell approaches, are just taking us one step closer to achieving or getting to that holy grail that we've been talking about.
What was the term that you called it? Kind of teaching the cells?
Yeah, retraining the immune system is what I call it. It is really trying to retrain the immune system because immune system evolves to fight against anything foreign to us. That's how we can survive infections. That's how we survive cancers because what is not ours is foreign and we have to fight it. That's how the immune system works. If we can retrain it not to react to just specific proteins that are on the surface of that organ without changing its reactivity to infections or cancers, that would be the tolerance.
If we could look through a microscope, what happens when you introduce some of these donor cells to the recipient and they teach the other cells? How does that work?
Perhaps I can address that because our studies are specifically focused on infusing donor-derived cells in the context of living donor liver transplantation. We believe that these cells, when they are infused into the body, actually don't survive very long, but they pass on messages to the recipient immune system. And these messages are, we believe, conveyed in the form of small particles or vesicles that are derived from the infused donor cells. And these particles, they're sometimes called small vesicles, extracellular vesicles, they convey the donor antigen, but in addition, they also convey instructions, protein molecules, and possibly also RNA molecules that when they are accepted by the recipient immune cells, condition those recipient immune cells to be unresponsive specifically to donor antigen because the donor cells that were infused express the donor antigen. And studies that we've done for many years at the University of Pittsburgh do indicate that this is how the cells are instructing the host immune system to be less responsive or unresponsive to the donor organ.
When all this happens, when all these messages are received, what does the liver then do next? Or how is the liver sort of a clue that this is effective?
Yeah, liver has a way of retraining the immune system itself. We have done several studies here at Mayo Clinic showing that the liver itself, after the transplant, changes the immune responses against it through working with the recipient immune system. That's how it tends to protect the simultaneously transplanted organs from rejection. When you do a liver and a kidney transplant, the incidences of rejection in the kidney are just much, much lower despite the same immunosuppression. So we know that liver has that capacity. Achieving that, as Dr. Thompson said, would allow us to just reduce the immunosuppression very controllably, slowly, making sure there is no injury to the liver, and eventually to completely get rid of the immunosuppression that people have to rely on. I think each of us are making progress through our own unique ways, trying to retrain the immune system. As I said, at Mayo Clinic, we have several upcoming trials in liver transplant patients. But in addition to that, something we haven't talked about, we have started aiming at other organ transplants as well. Just because liver seems to be a little easier should not lead to just working on liver transplant patients, because everybody else also needs this kind of innovative approach. So we have infused cells into the kidneys of kidney transplant recipients with rejection to slow down the rejection using or mimicking the biology, using some of the anti-inflammatory cells to help us reduce the inflammation with it. That those have been successful thus far as well. And just like Dr. Thompson said, we are at the phase of expanding those trials to do more efficacy studies.
Dr. Thompson, what is the potential, as Dr. Tanner just mentioned, of other organs? He mentioned kidney. What other organs could this be effective with, or what are you studying there?
I think we have to recognize that while the spontaneous occurrence of transplantation tolerance is relatively common in liver transplantation, it is very rare, exceptionally rare in kidney transplantation, but it does occur. So these natural occurrences of clinical transplantation tolerance really encourage us to pursue the cutting edge approaches that we believe, a step at a time, will take us closer to the holy grail of transplantation tolerance. So, kidney is clearly amenable to many of these innovative approaches to conditioning the organ or conditioning the recipient with innovative therapeutic agents, whether it's cell therapy, new biologics, or new pharmacological agents. The heart and lung, where perhaps the greatest unmet need, these organs clearly the patients would benefit from the same advances. And although it is more challenging, it is still possible to think about applying these approaches in thoracic organ transplantation.
Thoracic organ transplants, hearts and lungs, are life-saving transplants. They too need to be on anti-rejection medications because the consequences of having a rejection in those organs are quite high. Then they are kept on higher doses of immunosuppression. So it would be great to be able to take these innovative approaches to the thoracic organs, as Dr. Thompson said. And I'm hopeful that we're getting closer and closer to that point.
I love what you said, that transplant medicine has become so refined and sophisticated over the past 50 years that in solving one problem, we've created another. And now it's your moral imperative, really, to solve this other problem with immunosuppression medicine. Is that how you see it, Dr. Thompson? This is the new frontier for you as an immunologist?
Yes, as an immunologist, my career long goal has been to ultimately improve outcomes for the transplant patient, reduce dependency on anti rejection therapy, and getting closer to the goal of clinical transplantation tolerance. We have a recurring, very real issue of inadequate supply of donor organs.
That's a real issue, and that has been a real issue ever since transplant has started. We simply do not have enough organs for people who are in need of organs. So a lot of progress on that has been made also. Part of that is thanks to the advances made in immunology, as we've been talking about. And part of that is trying to find other sources of organs like xenotransplantation, genetically modifying non human animals with similar sizes to humans to make them more suitable for transplantation. There's been a lot of progress in that in the last few years, too. There are a lot of immunological issues that goes with that, but most of those issues have been tackled now compared to where we were 20 years ago, and that is becoming a reality in the next few years as well.
So xenotransplantation. So that's organs that are sort of modified from animals, like you said, like pig.
Correct. The most common source is pigs because pigs have very similar sizes of their organs as humans, so that makes sense. And genetically modifying these pigs to make them more similar to the human organs. The initial trials are underway to start probably with kidney and heart transplants. You might have heard or read the news about those things. The larger trials are coming in several centers, including Mayo Clinic, that will involve trying these organs in patients who are in desperate need of organ transplantation.
I'd love to bring it back to the patient. I hear a lot of excitement and where this is headed, but also a lot of careful optimism that you have to take the next step and the next step. What have we learned so far from the patient journey? I know that there are still many patients out there trying to stop medications after years of what's known as tolerance focused therapy.
The willingness of patients to participate in promising clinical trials is very precious. And as clinicians and scientists, we are very grateful to patients and their families for participating in these studies that offer, as Dr. Tanner has said, considerable grounds for optimism. And as transplant scientists, I feel, are entitled to be optimistic because of the incredible strides that have been made. And I think that inspires us, it inspires patients, and it inspires families to have optimism about the future of the field.
Dr. Tanner, what do you hear from patients that have received organ donations and what do they face?
Transplant recipients are very grateful to their donors. The first thing is that we cannot do any of this without the donors. So a lot of credit goes to donors, whether they are deceased donors or living donors. That is truly a gift of life. So that's number one. Number two, most recipients really are very proud of the process they went through to get a transplant, and they want to keep that transplant as long as it goes forever. And that's our goal as well. So our job is to help them do that. So all these efforts to try to reduce the immunosuppression, to get them off the immunosuppression, is to help them get to that healthy life and keep that precious gift as long as possible.
Dr. Thompson, there is another link between what we're talking about today with transplant tolerance research, but also autoimmune conditions like diabetes, type 1 diabetes, inflammatory bowel disease, and rheumatoid arthritis. So I'd love to hear how what you're learning now can also have a lot of potential with autoimmune disease treatment as well.
Those of us who perform science in transplantation and perform science in the many autoimmune diseases share an understanding of mechanisms that prevail in both conditions. The immune response is an undesired phenomenon in transplantation and in autoimmune diseases. And the two fields do learn and are instructive for one another. The development of immunosuppressive drugs in the field of transplantation led to their application in numerous autoimmune disorders. And similarly, the successful trials of immunosuppressive agents in the field of autoimmunity can be instructive for transplantation. There have been quite a number of examples of this two-way relationship. One field learns from the other. Reagents that may prove new agents that may prove to be very effective in controlling certain autoimmune disorders may be valuable for testing in organ transplantation. But overall, there are many mechanisms that are similar between the rejection of an organ transplant and an immune-mediated autoimmune disorder.
Yes, I completely agree with all that. So immune system plays a major role in a lot of disease processes. So while transplant is allowing us to study this in a sort of a man-made disease condition, the findings are helping other patients like those with autoimmune disorders.
I'm just curious, when you look decades into the future, what research implications are there beyond today's transplant patients? I'm thinking about lab-grown organs, gene-edited organs, or other technologies that we're just beginning to explore.
Those are all on the table, and a lot of people are working on different parts of this whole progress. In the future, we should be able to grow organs or parts of the organs in the lab. We should be able to adequately modify the genetics of animals or lab animals that we can take their organs from and transplant into people. Hopefully, the field will look much different in the next 30, 40 years as it looks different now compared to 30, 40 years ago. I think the progress has been very exciting in this field.
Dr. Thompson, yeah, if these advances continue to move forward, what does this mean for patients and families living with the realities of transplant today? And what do you hope for? What do you imagine?
I think these advances instill enhanced optimism about continued progress. There's a remarkable shared vision between clinicians, scientists, and donors, graft recipients, and their families. And this shared vision is something that I personally feel really does help sustain the field. So, if we think a generation ahead, I believe we will be less dependent on the conventional anti-rejection drugs that we've been dependent on for so many years. And we will be using approaches that have less side effects, cumulative side effects over time.
Thank you so much. And Dr. Tanner, as Dr. Thompson was speaking, I was thinking just what promise or what hope is on the horizon as a gift to your patients who have really been through so much, to be able to tell them there is something around the corner has got to keep you hopeful as well.
If you take a step back and look at the transplant process, you realize that how much progress has been made over the past 50 years or so. I would tell the patients that the progress is going to continue. Thanks to the donors, we're able to do all these studies, adding scientific data to their care. And this is all ending up with better and better graft survival, better patient survival. And we're very optimistic about the future in transplant medicine.
In our podcast, Tomorrow's Cure, we really focus not only on innovation, but that grounded sense of hope. And I'm just wondering where you're at with your studies and your research. What gives you hope and maybe a little bit of wonder about the future of transplantation?
I think that the history, the developments, and the shared optimism about the future are what will continue to drive this field forward.
Yeah, Dr. Tanner, it really is this ecosystem of care all the way from the person who decides to give their organ all the way to this moment.
This all goes back to transplanting being really a team sport. There are some things in medicine that involves just the patient and the treating physician. Transplant is not one of them. Transplant is a unique treatment in medicine that involves two individuals, really connects two individuals, the donor and recipient, comes with a whole team that supports the success of that transplantation. And that unites the team, including clinicians, the basic scientists. That gives us hope that this will continue to improve and the outcomes will be much better than what they are now, which is still fantastic.
Wonderful. Well, thank you both so much for your history of collaboration, for your research, and for enlightening us today. I really also approach this field with hope for all the patients who have been through so much. So thank you. Thank you again.
Thank you very much, Lindsay.
Thank you. It's been a privilege.
Tomorrow's Cure is a production of Mayo Clinic with production help from The Podglomerate. Be sure to follow Tomorrow's Cure wherever you get your podcasts. And if you liked today's episode, please like and subscribe. I'm Lindsay Sievert. Thank you so much for being with us.